we'll be talking about the properties of cardiac muscles. Now, before we proceed, let us talk about the excitability of cardiac muscles. Now, excitability is defined as the ability of living tissue to give response to a stimulus. In all the tissues, initial response to a stimulus is electrical activity in the form of action potential. It is followed by mechanical activity in the form of contraction and secretion. Now, let us look at the electrical potentials in cardiac muscle. The resting membrane potential in a single cardiac muscle fiber is between -85 to -95 millivolts. The resting membrane potential in sinoatrial node is between -55 to -60 millivolts. The resting membrane potential in Purkinje fibers is between -90 to -100 millivolts. Now, the action potential in cardiac muscle is actually different from that of other tissues such as the skeletal muscles, the smooth muscles, and the nervous tissues. Now, the duration of the action potential in cardiac muscle is 0.25 to 0.35 seconds. Now, the action potential in a single cardiac muscle fibers occurs in 4 phases. Number 1 is the initial depolarization. Number 2 is the initial repolarization. Number 3 is a plateau or final depolarization. And number 4 is final repolarization. The initial depolarization is very rapid and it lasts for about 2 milliseconds. The amplitude of depolarization is about plus 20 millivolts. Immediately after depolarization, there is an initial rapid repolarization for a short period of about 2 milliseconds. The end of rapid repolarization is represented by a notch. Afterwards, the muscle fibers remains in depolarized state for some time before further repolarization. It forms the plateau in action potential curve. The plateau usually lasts for about 200 milliseconds in atrial muscle fibers and for about 300 milliseconds in the ventricular muscle fibers. Now, due to the long plateau in action potential, the contraction time is also longer in cardiac muscle by 5 to 15 times than in skeletal muscle. Now, final repolarization occurs after the plateau. It is a slow process and it usually lasts for about 50 to 80 milliseconds before the re-establishment of resting membrane potential. Now, let us talk about the ionic basis of action potential. Initial repolarization, which is the first phase, is because of rapid opening of fast sodium channels and the rapid influx of sodium ions, as in the case of skeletal muscle fiber. For initial repolarization, the initial repolarization is due to the transient opening of potassium channels and efflux of a small quantity of potassium ions from the muscle fiber. Simultaneously, the fast sodium channels close suddenly and slow sodium channels open, resulting in slow influx of low quantity of sodium ions. Now, the plateau is due to the slow opening of calcium channels. These channels are kept open for a longer period and cause influx of large number of calcium ions. Already, the slow sodium channels are opened, through which slow influx of sodium ions continues. Because of the entry of calcium and sodium ions into the muscle fiber, positivity is maintained inside the muscle fiber, producing prolonged depolarization, which is the plateau. Now, calcium ions entering the muscle fibers play an important role in the contractile process. The final repolarization is due to the efflux of potassium ions. The number of potassium ions moving out of the muscle fiber exceeds the number of calcium ions moving in. It makes negativity inside, resulting in final repolarization. The potassium efflux continues until the end of repolarization. Now, at the end of final repolarization, all the sodium ions which have entered the cell throughout the process of action potential moves out of the cell and potassium ion moves into the cell, and this occurs by activation of sodium-potassium pump. Simultaneously, excess of calcium ions which have entered the muscle fiber also move out through sodium potassium pump. Thus, the resting membrane potential is restored.